email Andrew and see if he can get on.
I think he is.
Yeah, you don't see me?
Oh, you're there?
Hey,
No, I don't.
how's it going? Hi, Emily.
Hi, Andrew. Good to see you.
Good to see you too. I had to switch to my phone because my laptop is low on memory and it keeps crashing. So.
I was like,
anyway.
oh, dude, what's going on in your nose there?
Yeah, sorry.
So we're good.
Fun. Yeah.
All right, so
I'll just turn that camera off.
My name is Stanley Grant, and I'm director of the National Science Foundation Partnerships for International Research and Education, or PIRE program, here at UC Irvine. And today I'm very pleased to introduce Dr. Emily Payne, who's going to talk to us about some of her nitrate removal research in biofilters at Monash University. Emily received her BS, Bachelor's of Science in Environmental Engineering and Science at the University of Melbourne in 2005, and then went on to get her PhD at Monash University in the water group within civil engineering at that university, and she finished her PhD in 2013. Currently, Emily is a research fellow at Monash University and also part of the CRC for water-sensitive cities within civil engineering. And her work on biofilters is quite seminal and widely cited, so we're very, very lucky to have her present to us today. So without further ado, Emily, I'm looking forward to your presentation.
Thank you so much, Stan. And thank you very much to everyone being here today. I very much appreciate your time and being here to listen to my presentation. So I'll just bring it up on my screen, and I hope you've all been able to access it. So today I'll be speaking about nitrate removal from stormwater runoff in biofiltration systems. And this work really comes from my PhD, but I also worked closely alongside a master's student, Tracy Pham, and my supervisors were Tim Fletcher, Perrin Cook, and Belinda Hatt, and Anna Deletich was also closely involved in the project, so I'd really like to acknowledge all of their input into this work. So moving on to the next slide. Nitrogen cycling has been widely studied across a range of natural and modified environments, but biofilters have some unique conditions that can influence nitrogen cycling within them. So these systems aim to harness natural treatment processes, but within an engineered environment. Inflows are highly variable, so drying is a key and expected part of stormwater biofilters. And as a result, we usually use terrestrial plant species within these systems. The filter media has a very low nutrient content from the outset, and that has been found to be important to prevent nutrient leaching. But it creates a really barren environment for the plants at the beginning, and they're expected to receive their nutrients from the incoming stormwater flows. So slide 3 just shows the plants, the filter media, and the microbial community. They all play an important role in nitrogen cycling within these systems. So on slide 4, just looking at the nitrogen cycle within these systems, nitrogen removal is complicated by the various inorganic and organic forms of nitrogen, the different transformation processes that can occur, and also the potential for nitrogen to be recycled via the plants and then decomposition processes that act to re-release nitrogen. So some key processes in the nitrogen cycle are uptake or assimilation by plants and microbes, decomposition and then mineralization processes, which re-release inorganic nitrogen from organic forms of nitrogen. Then there's nitrification, which requires aerobic conditions, and then denitrification, which provides a permanent removal pathway, converting nitrate into nitrogen gas, but it requires contrasting anaerobic conditions. So on slide 5, nitrogen removal from these systems has been shown to be highly variable, with studies showing sometimes showing effective reductions in the order of a 70% reduction through to leaching of nitrogen. And in particular, nitrate is particularly challenging to remove. It's highly mobile, and so it readily leaches from these systems. And it also can be produced by nitrification, which happens quite efficiently in the aerobic portion of these systems. So on slide 6, just looking at some factors that influence nitrogen removal. So studies have shown that performance is sensitive to, and if you just click through these, the presence of plants. Many studies have highlighted the much poorer performance from non-vegetated systems. And then the media nitrogen content, which I mentioned. So it's very important to have low nutrient media to prevent bleaching. Plant species selection is also an important factor for the performance. A study by Jenny Reed in 2008 found that only 1/4 of 20 species that were tested had more effective reduction of nitrogen compared to the non-vegetated controls. Many studies have also shown the importance of a submerged zone and a carbon source to provide the conditions available for denitrification. And finally, removal is also sensitive to drying. So we see much poorer removal after a very prolonged dry period. So on slide 7, we conducted a literature review to look at nitrogen cycling within a range of environments and to try and draw some implications for nitrogen processes that we could expect within stormwater biofilters. And some of the key findings to come out of this literature review were the importance of plant and microbial interactions. So the plants and microbes have very close interrelationships with each other. Most of the microbes exist in the rhizosphere, the zone immediately surrounding the plant root. And the microbes receive carbon-rich exudates from the plants, and in return the microbes provide benefits to the plants, helping to re-release nutrients for plant uptake. And it follows that the root architecture is also important. So as roots populate the filter media, that influences the way that stormwater that filters through the media will come into contact with the plants and also with their associated microbial community. Plant strategy is also a factor that can influence the way different plant species will interact with nitrogen cycling. So different species will vary in their biomass allocation, their composition of tissues, and also in terms of the strategy they employ in terms of their investment of resources. So different species might employ a strategy of slow growth where they construct tissues that are robust, heavily defended, relatively dense tissues that tend to be longer lived, but they'll be slow growing. Whereas other plant species might go for a rapid growth strategy, but that comes along with tissues that can be relatively short-lived and more readily broken down. And so studies have related these different strategies to fast or slow nitrogen cycling within systems, and different strategies will benefit different plant species depending on the environment. Also, the distribution of moisture throughout these systems is important to influence the different processes that can occur, the mobility of nitrogen throughout the biofilter, the capacity for plants to uptake nutrients, and processes such as the conditions available for nitrification and denitrification. And even moisture heterogeneity on a micro scale will influence, say, the coupling of nitrification and denitrification. So on slide 8, I've just got a bit of a bar chart that shows the breakdown of different forms of nitrogen within the influent, the stormwater influent. And this is the influent we used in our study, but we were really trying to target the stormwater composition that has been found from field sampling studies. So here I really just wanted to show that nitrate forms a really high proportion of the inflow, up towards 45% or so. And if you just click to highlight to show the breakdown of different forms of nitrogen that we saw in the different outflows from the different designs we tested. So here we tested free draining designs and then designs with a submerged zone. We simulated wet and dry conditions, and we had vegetated and non-vegetated controls here. So if you just click again to highlight the fraction of nitrate, I really just wanted to show here that it also forms quite a dominant proportion of the outflow from the biofiltration systems. And particularly the nitrate forms a dominant fraction in the non-vegetated systems. So it really just highlights the challenges of nitrate removal within biofilters. So moving on to slide 9, This photo shows the column experiment that we undertook. We were aiming to get a better understanding of nitrogen processes and do some further testing of the way that different designs and plant species impact performance. So we tested 22 different plant species. Each of these we also tested free draining biofiltration designs versus designs with a submerged zone and a carbon source at the base of the filter. And we also simulated wet and dry periods, and this was over a 1.5 year period. And we measured various parameters over this time, and I'll present results on many of these. So slide 10 just shows a box plot distribution for nitrate concentrations from the outflow of our different plant species that we tested. So the different species are shown along the x-axis. Concentrations of nitrate in the influent are shown by the red line. And the boxes that are white represent the free draining non-saturated designs. Boxes that are blue are the biofilters that had a submerged zone and a carbon source at their base. And so these are the results from the wet period when we were dosing twice weekly with stormwater. And if you just click to highlight, the results really show that all plant species we tested were relatively effective at reducing concentrations of nitrate compared to the non-vegetated controls. So you can actually see some nitrate production from these non-vegetated controls, indicating that nitrification is occurring in these systems. So it really highlights the importance of including plant species in these systems. And it shows these results really show greater consistency in performance across plant species compared to earlier studies like the 2008 study by Jenny Reed. And this is because biofiltration media has become sandier with a low nutrient content over this time. So we use low nutrient content media, and it shows that you can expect greater consistency in performance across different plant species. So it means a wider range of plant species are appropriate for these systems. So just clicking onto slide 11, but what about the influence of drying? So drying is a key characteristic of stormwater biofiltration systems. We can expect drying. So how does that influence performance? Well, now slide 12 just shows down the bottom a comparison compared to our dry period. So our columns underwent up to 15 days of drying in the summer period, but that represented quite severe drying for them because we had small columns housed in a greenhouse, so they got quite warm. So as expected, we can see the reduction in performance that comes from drying. There's greater variability there between plant species, but you can now see the clear benefits of including the submerged zone. Now moving on to slide 13, these scatter plots just show the variation in water quality between water that's stored in the pore water or within the submerged zone in between storm events, and that's shown on the x-axis, so the old resident water, compared to the quality of water that passes immediately through the biofilter during an inflow event. So that's the new water, and that's shown on the y-axis. On the left, we've got some results from the free draining designs, and on the right, we've got results from designs with a saturated zone. So if you just click a couple of times to bring up the red circles highlighting the different results, we can see that, so the different symbols show the different plant species we tested, and individual columns for each symbol. And you can see that the most effective plant species had rapid treatment, so they have low concentrations of nitrate in the outflow, irrespective of whether it's water that's been stored between events or water passing immediately through the filter. So really showing how quick processing, this effective processing is occurring in these columns. But then other plant species were less effective and had slower processing. So the water passing immediately through the filter has a poorer quality compared to the water that's stored within a system between events. And it's very clear for designs with the saturated zone the benefits of retaining water in the system in between events, allowing time for processing, biological processing to occur. And in these columns you can see that given enough time, irrespective of which plant species is used, all of them can reduce concentrations of nitrate back to relatively background concentrations. And I should also point out these results are from the wet dosing period. So again, showing how the submerged zone can help to even out differences in plant species performance. So on slide 14, these are some results from the nitrate isotope tracer that we added to a subset of columns. So we really wanted to look at the fate of nitrogen within the system, and particularly look at the partitioning of nitrate between uptake by the plants and microbes, which really represents something of a temporary storage. At some point that nitrogen may be re-released upon turnover of the plant or death of the plant or microbe, versus denitrification, which converts nitrate into gaseous form and provides a permanent removal pathway from the system. So slide 15 shows the results of the isotope tracer addition. These different columns show the 3 replicates tested across different plant species, with the non-vegetated system at the far right. So the green shows nitrate that was taken up by the plants and microbes, blue shows the proportion that was denitrified, and yellow shows the nitrate remaining in the pore water. So we were very surprised to see that most of the nitrate was being taken up by the plants. Denitrification formed a relatively minimal removal pathway, and particularly for the most effective plant species such as the leptospermum and the juncus, we can see that most of the nitrate, almost all of it, is being taken up by the plants. And we think that the most effective plant species are taking up most of the nitrate in the upper layer of the biofilter before it's able to even reach the underlying submerged zone where the conditions for denitrification exist. So these results are from early biofilter life. So our columns were only up to 1.5 years old. So it would be interesting to see what kind of processes are occurring in mature systems. So slide 16 just shows some results from the plant harvest we conducted. So we conducted a plant harvest at the beginning of the experiment to characterize plant and initial biomass and root morphological characteristics, and then at the end of the experiment we conducted a huge harvest of all of our plants to again characterize their growth and morphology. So we related these plant characteristics to their performance for nitrogen removal, and we found results that are also similar to the study of Jenny Reed in 2010, where the most effective plant species had a really extensive root system. So high total root length, a high surface area of the roots, and a high root mass. Also a relatively high length of fine roots, and this extensive root system also tended to go hand in hand with a higher total plant biomass. Whereas the poorer performing species tended to have a limited root system, generally thicker roots. But it should be noted as well that there was some variation in species performance. So some species did well in the wet period, or didn't do so well in the wet period, but then did better in the dry period. And I think because they were able to conserve water due to their low growth and low uptake strategy. So it indicates that including a mixture of plant species is important, but the most effective performers across the board were differentiated by their extensive root system. So slide 17 just shows some conceptual diagrams of the processes that could be occurring here, looking at the influence of different species characteristics and the submerged zone on processing of nitrate. So the first diagram just shows a plant species with an extensive root system, so an effective plant species. Nitrate entering the system and filtering through the media is effectively taken up by assimilation in this root system, leaving little nitrate to come out in the effluent. But then clicking through to the next diagram, we can see a plant species with a poorer root system, less effective contact with the nitrate as it moves through the filter media, allowing more of the nitrate to pass through the system, in this case a free draining design. So less effective uptake. And then the final diagram shows that poor plant species, but then coupled with a submerged zone. So the submerged zone is then able to provide retention between events, allowing more time for uptake by the plant and also providing the conditions for denitrification to occur. So that results in better performance and reduced concentrations of nitrate in the outflow. Just considering the role of soil organic matter before we finish up. So the results from the isotope tracer, which really show the importance of plant uptake, really lead to more questions in terms of long-term nitrogen cycling. So what will happen once the plants stop growing so rapidly and start to turn over more of the nitrogen we expect will be returned to the system. And perhaps denitrification will then become more of a significant pathway. But also, will a pool of soil organic matter start to accumulate within the system over time from the turnover of the plants and the microbial biomass, and also from organic inputs from stormwater entering the system? So in many terrestrial systems, the pool of soil organic matter can form quite a significant storage for nitrogen. One study reported that the soil organic matter can, the size of this storage can exceed the size of nitrogen stored within the plant biomass by an order of magnitude. So the soil organic matter will comprise a wide range of substances, some of which may turn over very rapidly, be decomposed and re-released quite rapidly, but other substances will be more recalcitrant and stable and provide long-term storage. So I think it will be an interesting question to see whether the soil organic matter, how it accumulates in these systems, and whether it can also provide a long-term sink for nitrogen. And different plant species will interact in different ways with the pool of soil organic matter. They have different composition. You can imagine woody plant species have more recalcitrant components, so that will be an interesting question as well. And I think that in agricultural systems, soil organic matter and the benefits of it are well understood, but it's an interesting question for biofiltration systems, particularly given at the outset the importance of having a low organic content in the media, but we don't know much about its accumulation over time and whether it can benefit or perhaps be detrimental to performance. So just finishing up on slide 19, this just is a summary of some of the factors that are important in designing these systems to ensure you can optimize nitrate and therefore total nitrogen removal from these systems. So again, having the sand-based media with a low nutrient content to reduce leaching, including the submerged zone and the carbon source, to provide the conditions that are required for denitrification, and also provide that prolonged retention which allows time for slower processes such as plant uptake to occur, and it helps to reduce the detrimental impacts of drying on the performance of these systems. Plants are also essential for nitrogen removal, and that's well acknowledged. So it's important to include a mixture of species within these systems, but also to include amongst these species that have extensive root systems, relatively high biomass and growth, to really allow effective nitrogen uptake and know that the plants are supporting an extensive microbial community to contribute to processing within these systems. So just finishing up on slide 20, thank you very much for your time. I very much appreciate you listening to my talk today. Thanks very much.
Thank you, Emily. Yeah, absolutely. Wonderful, wonderful talk.
Very kind.
So questions for Emily?
I have a question. I can't see everybody else. Is this okay to ask?
Yes, please go ahead.
Okay, I wanted to ask for a little bit of clarification about the last point about organic matter buildup. I mean, well, two questions. First, do you know whether the nitrogen preferentially accumulates in the leaves or the roots, or is that species dependent? And then the other question is, once all of that ends up in the soil, doesn't remineralization actually release it into the outflow eventually?
Yes, that's definitely the case. So your first part of the question, look, I'm not sure. I think it is species dependent in terms of the allocation, and I think even plant species will vary in terms of how much they allocate above ground versus below ground. So that will be quite variable between species. Absolutely, and that recycling factor does really complicate the removal of nitrogen. I think it's something we don't know enough about. There's certainly the potential for re-release, and that will occur, and re-release and then subsequent leaching. But I think we don't know whether the nitrogen can be re-released and then either re-taken up by plants or denitrified, or whether some components will be those recalcitrant, difficult components that are difficult to decompose, and whether there will be some sort of long-term storage there. But certainly I think a lot of the nitrogen will be quite rapidly broken back down and re-released, and then potentially could be leached from the system. So that is a real risk there.
So it might be better to harvest them.
Yes, and that's certainly some work that Brandon looked at. Yeah, and it's a big question, and I think you're right. It could be a double-edged sword. I just really wanted to raise the question of there's not enough known about the accumulation of organic matter because it will be inevitable to some extent. Certainly the recycling and re-release is a threat to the performance of these systems, but I just was interested in whether there could be some storage of sort of the more recalcitrant compounds there.
So Emily, I have a sort of follow-up question from Lisa, and maybe two parts to it. First, you talked about assimilation, including the microbial community in your preamble, as well as the plants. But then when you shared your results for the N15 labeled nitrate studies, it looked like the assimilation was plant-based only. And I just kind of wanted to probe you on that a little bit and find out if that means that the implication would be that the microbial community was not assimilating. And then the second part of the question has to do with timescales. So, of course, if you run an experiment over a particular timescale, you only interrogate processes that occur over that timescale. I'm just wondering if you could tell us a little bit about how you ran the experiments and whether there could be, like for example, if the rate of denitrification was much slower and then the timescale of the experiment, then you'd sort of expect, but the assimilation, plant assimilation was much faster, you'd sort of expect that you might see a bias in your results toward that. Can you just kind of clarify how all that works? It's a very complicated system, so
yeah,
looking forward to hearing what you think.
Thanks, Stan. Yeah, so the first part of your question, the microbial assimilation, you're right, that is important, and I did tend to, on my slides, just talk about the plant assimilation. We didn't explicitly differentiate plant and microbial assimilation, but from the results from the non-vegetated control columns, you can see some assimilation there, and it's quite significant proportion. So that does show that I think the microbial community is assimilating the nitrate. And it's certainly important alongside the plant assimilation. So we ran the isotope test. We input the nitrate tracer with the stormwater, and then we monitored concentrations of nitrogen gas within the pore water immediately within the system, so as the stormwater drained through the system, and then over the following days until the next stormwater dose was applied. So in the wet period, we were monitoring over about a 5-day period, and then in the dry period, it was about over a 14-day period of drying. So it was sort of looking at the dynamics over between one inflow event to the next. And I think you have a very interesting question, but we plotted the increase in the 15 nitrogen gas, and we showed quite a rapid rise there, and we used that to calculate our rate of denitrification. I think when we did our mass balance, we calculated it over a 24-hour period, I think. So we really did try to capture, we did more rapid sampling at the beginning and tried to capture what was occurring initially. So we saw that the nitrate was quite rapidly being denitrified if this process was occurring. I hope that answers your question.
Yeah, so just as clarification, then it really is about sort of starving the saturated, well, the fact that the plants are located higher up in the column, so they're capturing the nitrate before it gets a chance to, so the denitrification is nitrate limited, I guess, would be one way to look at it.
Yeah, I think so, and I think that's why we see the poorer performing species are associated with a higher denitrification proportion. Yep.
Beautiful, thank you.
Thanks, Dan.
So I have a question, a couple actually, about the saturated zone. So what do you know about the depth of the saturation zone and how far should it be from the surface, and how deep should it be, and sort of how does that interact with plant roots potentially?
Mm-hmm. So the saturated zone, we recommend it's in the lower portions of the media, so not within the upper filter media, but within the sand transition layer and the gravel drainage layer below. And I think this stemmed from earlier studies that showed leaching from that filter media, particularly if it was saturated, it could re-release phosphorus. But the filter media has a lower nutrient content now, so I suspect it wouldn't be as much of a problem. But it's really the lower layers of the media. We were surprised when we did our final harvest that most of the roots were concentrated in that upper layer of the filter media, the unsaturated zone. I really expected more roots to be throughout the column, and it definitely varied between different plant species. But I think the results do show that plants were able to access the nitrogen retained within the submerged zone following between events. And clearly the submerged zone helped them to survive and maintain better performance following the dry period. So I think that the roots were interacting with the submerged zone, even though they were predominantly located above.
Yeah, so that's actually a surprise that they're predominantly high up.
Yeah. I think it's because we had our column experiment during the wet period. They were receiving very regular inflows, so I think it probably only encouraged surface growth of roots. So it's a bit of an artifact, I think, of our column experiment.
Yeah, so because here in our Mediterranean climate, where we have 8 months of a dry period, we're interested in how that might affect the performance, both in terms of perhaps the survivability of the plants, although maintaining a saturated zone would be a lot more challenging. But also whether that might be different if we have a longer dry period. So actually that was my second question, was about the dry period. And so your dry period was it 14 days, did you say? Or 10 days?
Yes, up to 15 days, and I know that's not very long.
Not for us, yeah. And so any ideas about how the functions would change with a longer dry period?
Yep, so it's certainly not even a long dry period for our climate, and particularly certain areas in Western Australia. It was partly because we had such small columns, and we were trying not to kill our plants before the end of the experiment, so we had to really walk a fine line. I think that they experienced much more severe drying relative to the bulk soil of a field system over that 15-day period. They were in quite a hot greenhouse with a concrete floor, I think the results could be extrapolated for a longer dry period, but definitely we recommend submerged zones in all systems over here, particularly if you experience over 3 weeks of drying, and it will help performance, but it also reduces the maintenance requirement in terms of helping to water the system through dry periods. If it's a very prolonged period, you would need to look at topping up the submerged zone. But again, that should be a lower maintenance burden compared to a system that's really struggling without a submerged zone, particularly with the really sandy media now. It's so important to provide that for plant survival. I hope that answers your question, Richard.
Yes, it does. Thank you.
Thanks.
I have another question about some data that I just sent everybody on email, but I'll just describe it to you, Emily, and see if you have some ideas. We have a biofilter right along the ocean. We have several that were monitored in our rains this year. It's kind of unusual for us to have rain. But anyway, there are 2 that we monitored, and they both took up a lot of the incoming ammonia. Mm-hmm. So there was a significant reduction in ammonia in the outflow, but a significant increase in nitrate in the outflow. And in the case of phosphorus, it was one of the filters had more phosphorus coming out, and one filter had more phosphorus. It had a reduction in phosphorus coming out. Mm-hmm. So I'm just curious, what do you think would cause the ammonium uptake but nitrate release? And I have to say, there's not much vegetation on these. There's just a little bit.
Yep, I would say that you've got really efficient nitrification occurring in the system, and nitrifying bacteria, they're effective even in the non-vegetated systems, so that's converting the ammonium to nitrate and increasing your nitrate concentrations. But the nitrate removal really is more heavily dependent on the presence of vegetation, either from uptake by the plants or really if you have a submerged zone there and there are conditions for denitrification, the plants also help provide carbon for denitrification to occur. So it sounds like with a non-vegetated system,
we're just having nitrification, right?
Yeah, I think it looks like it. You don't have that next step to remove the nitrate, either by plant uptake or denitrification.
And so, yeah, okay. So ideally, if we had a lot of plants there, all that nitrate would have been taken up and not show up in the outflow.
You should see an improvement, yes, yeah. And then if there's a submerged zone, again, you should see even further improvement, I think. Yeah, and the phosphorus.
Fast?
Sorry?
That process is fast?
Denitrification. Nitrification. Yes, I think it is. It is quite efficient. Yeah, and it happens within the aerobic portion of the system, so it seems to be quite an efficient process. I think that for the phosphorus, that's quite heavily dependent on the media, so a lot of phosphorus will absorb to the media, but it also can be removed by plant uptake, so it's a bit difficult to say why one system would have effective phosphorus removal and one wouldn't, unless there might be a difference in the media between the two systems perhaps, or whether one system has more plants taking up the phosphorus.
But one is producing phosphorus, so what would produce phosphorus? Did you see the video I sent? Can you see it?
Oh, I haven't opened it up yet. I'd say it could be release of phosphorus from the media, from some sort of source within the media, perhaps. So perhaps if there's a difference in some sort of composition of the media between the systems, that could be releasing phosphorus. Oh, yeah, I can see the results now. Does anyone else have any thoughts on that?
Andrew?
I guess the only other thing I would question about this is I'm not sure if the first flush of pollutants was always captured in the way the monitoring was done with these data. And so a lot of times I think that the influent pulse may have been missed. So I'm not sure if I would put too much faith in these data without additional monitoring. But yeah, I mean,
she said she tried to catch the first flush for a set of rain events for each set of events.
Yeah, I think that was the goal, but I'm not sure how much it actually happened.
Yeah.
But I think that aside from that, though, yeah, I mean, leaching from the media is possible. So I think that from talking to Kimberly, when they installed those biofilters, they didn't actually use the media that she had originally wanted. And there may have been some things as far in the specifications where the organic matter was higher and a number of other things were kind of out of whack, and that's why they're about to refurbish them.
Yeah, I mean, the outgoing phosphorus is pretty constant, so I wonder if it's independent of what's coming in.
Yeah, it's interesting.
So Emily, just a follow-up question there. When you report concentrations in the effluent, are these are they event mean concentrations, or I mean, are they flow weighted? I guess would be the right question, or are they are actual just concentration, average concentrations?
Yeah, I should have clarified that. Yeah, it's just an average concentration. We captured the entire outflow, and then took our sample from that. Apart from the results I showed, which took a concentration from the resident water versus the water passing straight through, that's when we subsampled the outflow hydrograph.
Got it, thank you. And so I just wonder, coming back to Lisa's question, if it would matter sort of whether you were doing flow-weighted monitoring or not. I always worry if there's some storage in these systems that what you call the influent concentration and what you call the effluent concentration don't really match up if there was a way to tag the water. So
yeah,
that's
I think
danger, I think.
Yeah, it's an interesting question. We tried to get a better understanding of that using a salt tracer through the system before we did the isotope study to really sort of characterize how much water is being retained. But yeah, definitely, I think the flow weighted results, give a lot more information. We were just limited by the number of columns we were trying to test as well. It's a very good consideration, I think.
Yeah, no, I think residence time is kind of my my favorite thing. So I think having an understanding of the residence time and sort of knowing what that delay time was, or just what the relationship of the residence time in the biofilter to what you're calling the influent and effluent concentration is pretty important. If you're talking about like a removal or an addition, because it could just simply be that you've aliased the temporal variability of the inflow and outflow.
Yeah, I think it's a very good consideration.
Emily, I had a question. Kind of the opposite of dry conditions. I was wondering if you had an exceptionally large storm, where you had, fairly, the biofilter was inundated with inflowing water, and then it just continued flowing because you had so much coming in. Do you think that assimilation by plants is fast enough to be able to capture the nitrate coming in the stormwater under those conditions?
Yeah, I think that's a really important question. I think the results where we sampled the water passing straight through the system give some indication. I think that the most effective plant species, yes, would be able to take it up rapidly enough, but I'm not sure to what extent they could maintain that uptake capacity throughout the duration of a very large storm. But yeah, I think you would see greater variation in the plant performance. The poorer species, no, they wouldn't be able to probably make much difference in that case. It's a very good question, though.
Okay, thanks.
That's a really cool idea that you could have a series of sort of rate limiting steps, and initially it might be mass transfer limited when the plant was really wanting to suck up the nitrate. But at later stages, actually, the plant, it could become biologically rate limited in the sense that the plant is kind of done. It's like, thank you, I've had my nitrate fill.
I'm full.
I'm full, exactly.
Yeah.
So how is that? How does that work? how would that be potentially quantified? So when the full of nitrate. Plant physiology lesson 101, I guess.
Yeah, probably is. It's a very good consideration. Perhaps you could add an isotope tracer throughout a longer period, but yeah, difficult, I think, to differentiate. It's a very good question. I think we need to understand it a bit more, the dynamics of the uptake and how long it's stored for, and then what happens when it's re-released. There's a lot of questions there, I think.
Mm-hmm. That's cool. Thank you.
Thanks, Dan.
Any other questions from anyone around the table?
I had a question about the microbial community, and what do you know about the microbes? Who might be the important players, and sort of what they might be doing?
Yeah, well, we're actually doing a bit of work in partnership with Cornell University at the moment with our microbial group, trying to look at the identity of which microbes are there, and then also look at the activity of the microbes. So really macrogenomics, I think. Oh, I can't think of the name properly. But yeah, we took some samples during our final harvest of the filter media, and we're looking to try and extract the microbial community from these samples and really try and get a bit of a better handle about the profile of the microbial community, such as whether the denitrifiers are present, and then trying to also look at the genes that are active there. So it's an interesting area, and hopefully there'll be more known about that in the near future.
Yeah, that sounds really interesting. And then what about the spatial distribution? Did you sample close to roots in the rhizosphere or away and high in the column and low?
Yeah, we did sample up in the upper layer of the filter media where most of the roots were found, and then in the lower portion of the biofilter within the submerged zone. But we did have great difficulty extracting enough of the microbial DNA from the lower samples. I think there wasn't much there. So I think the guys in the lab here have done a wonderful job coming up with new methods to try and extract this DNA, and we've got it for a few samples, I think. But we took relatively sort of bulk samples compared with sampling the rhizosphere zone. But I know another postdoc in the group did look at sampling that rhizosphere explicitly. That's a great question.
Yeah, because it seems like a big black box right now, so it'd be great to get a better understanding what's happening there.
Oh, definitely, definitely. Yeah.
All right. Well, it looks like maybe we've peppered you with enough questions, perhaps. Thank you so much, Emily. That was a really, really nice presentation, very concise and also very deep. So it was wonderful to have you present for us and answer all those great questions. And I guess unless there are further questions, we'll thank you and move on to the next set of presentations, at least in the larger symposium. So thank you again, Emily.
Thank you very much.
Yeah, that was really great. Thanks very much.
Thank you so much. I really appreciate your time.
Thanks,
Emily.
All right.